Good morning everyone. Welcome back to my channel Pari's tutorial. I'm back with one more interesting video. Please let's get into the class. Good morning everyone. Welcome back to my channel. In the last class I've already discussed about the glucose structures, carbohydrate structures and all, the 6 carbon compound, the structure. Start a detailed description of the metabolic pathways that happen in a cell. Okay, so first of all, I want to talk about glycolysis, because this glycolysis is a process which happens in almost all the living organisms, in all prokaryotic cells and eukaryotic cells. This is a must growing process, okay? And this process occurs in the cytoplasm of the cell. Each and every cell has to break the glucose into the simpler compound. The glycolysis name itself says that lysis is a breaking and glyco is a glucose component. So lysis of your 6 carbon compound into pyruvate is what you call a glycolysis. Okay, so I will also explain you the detailed steps of glycolysis, including the enzymes involved in it and what is the net gain of ATP for one molecule of glucose in a glycolysis. Okay, so let's start. So I told you glycolysis is an energy conversion pathway in many organisms. So glycolysis is a sequence of reactions that metabolizes one molecule of glucose into two molecules of pyruvate, and the net production would be two molecules. I will let you know in the detail cycle. I will write every cycle with the enzyme involved. Okay, so although glycolysis and glyconeogenesis, okay, although glycolysis and gluconeogenesis have same enzymes in common, the two pathways are not simply the reverse of each other. Okay, the two pathways would never happen simultaneously in the cell. Okay, so this is these two pathways are important pathways. Okay, and they would never happen simultaneously. Okay, so what are the easy steps for understanding in glycolysis? All would think that it's very difficult for us to remember the steps and enzymes involved. If you can easily follow a small cycle from small kind of enzymes, what are the types of enzymes and what is the function of that particular enzyme, it would be easy for you to remember those steps. Okay, so for your understanding, I am telling you this glycolysis is completely divided into three different stages. In the first stage, it is called as stage 1, where the conversion of glucose into fructose 1,6 bisphosphate would happen. Okay, and this stage is considered to be the ATP investment stage, because instead of producing ATP in glycolysis, in the stage 1 you will be taking the ATP. Okay, so this is called as energy investment phase. Okay, in the second stage is second called as, which is an interconvertible stage. It's a small cycle that happens in the stage 2, and nothing but the conversion of fructose 1,6 bisphosphate into two 3 carbon compounds called glyceraldehyde 3 phosphate and dihydroxyacetone phosphate. Okay, and this is the stage 2. And let's go to the stage 3, which is called ATP production phase. Okay, where your substrate level phosphorylation would happen. Okay, first ATP is produced in the stage 3. I will let you know every step in detail, because these enzymes play a vital role in your exams also. Okay, now let's go for the stage 1, where I'll directly do it in the this itself. Okay, for easy purpose I am opening it, because I want every step to be written in detail so that you guys would be understanding it. Okay, so I told you the first step is the conversion of your glucose, which is a 6 carbon compound, into glucose 6 phosphate. So instead of by hearting this step, I want to clearly tell you what happens here. See, glucose molecules first have to enter the cell. Once they enter the cell, they should never go out, because the glycolysis should happen inside the cell. For that glucose to not go out, it is phosphorylated. What is phosphorylation? It's addition of a phosphate group. Okay, so what happens is when a glucose molecule enters into the cell, there are some channels, protein channels on the cell membrane of your cell that would trap your glucose and would never enter the never make the glucose to go out. Okay, so once the glucose is trapped inside, the glucose is immediately phosphorylated, means it is immediately added with a phosphate group. This phosphate group would give you a polar nature, and this polarity bound molecule cannot bind to the protein channels, and there is no chance to escape out from the cell. So this is nothing but a trapping of glucose. This is the first step where glucose is trapped within the cell. Okay, and I want to tell you this enzyme, this particular step is first of all catalyzed by an enzyme called hexokinase. So why hexokinase? Kinase is an enzyme which mainly acts upon the phosphorylation. They are meant for the conversion of a phosphate group or the transfer of phosphate group from one molecule to another molecule. Okay, so this kinase, what happens is, here I told you in this stage it is an ATP investment stage. This stage 1 is an ATP investment stage, where instead of producing ATP, you are making use of ATP and ADP is produced. Means this particular kinase would help the conversion of the phosphate group from ATP to glucose 6 at the 6th carbon. So for example, consider this is your glucose molecule. I am drawing it in a linear form. Okay, so 1 2 3 4 5 6. So if to the 6th carbon you are adding a phosphate group, this is called your glucose 6 phosphate. Okay, this is catalyzed by the enzyme called hexokinase, because kinases are meant for the conversion of the phosphate group from ATP, it's a high energy molecule, to glucose 6 phosphate. So here ATP is used and ADP is produced. Okay, this is the first step of your glycolysis. Okay, now what is the next step? Glucose 6 phosphate is immediately converted, and it is an irreversible reaction, and it is converted to its isomer, which is a fructose 6 phosphate. Okay, so this reaction is catalyzed by an enzyme called isomerase. You all know glucose and fructose are isomers. So the glucose 6 phosphate is isomerized into fructose 6 phosphate. Okay, and now after this step, again there is a phosphorylation happening, which is a second phosphorylation. Okay, and again here there is a transfer of a phosphate group from fructose, I will tell you this, there is a transfer fructose 1,6 bisphosphate. So what happens is, here it is again catalyzed by an enzyme called phosphofructokinase. So I told you kinases are meant for the transfer of phosphate group from one molecule to another molecule. Again it is taking ATP and it is adding that particular phosphate to the first position of your fructose. So here it is one fructose 6 phosphate, and this particular ATP, this kinase would take that particular phosphate of ATP and it attaches to the first position of your fructose. So what happens? For example, this is your linear form of fructose, 1 2 3 4 5 and 6. Okay, and this is your first phosphorylated first position, and the 6th position is also phosphorylated. So here also it is ATP is reduced to ADP, right? So here for every glucose molecule already 2 ATPs have been reduced. Okay, now after fructose 1,6 bisphosphate, okay, it is also phosphorylated, and now till this step it is called the stage 1 of your glycolysis. Okay, this is called the stage 1 of glycolysis. In the next step I will detailedly draw again in the next page, so that it would be easy for you to understand clearly. Okay, these are the enzymes that are involved. Okay, now this is the first stage. Let's go for the second stage. Hope you are clear with the with stage 1. Okay, okay, so I told you in the first stage itself, it's a phosphorylation that is happening an isomerization is happening and a second phosphorylation is also happening. So stage 1 is done. So let's go for the stage 2. What happens? The cleavage of fructose 1,6-bisphosphate into two 3-carbon compounds called the dihydroxyacetone phosphate and the glyceraldehyde 3-phosphate. These are the two 3-carbon compounds that are produced upon from the fructose 1,6-bisphosphate. Okay, so let's go for the stage 2. See, this is the fructose 1,6-bisphosphate that I have done new, and with the enzyme called aldolase, which is also called as aldol condensation, where you produce 3 molecules of glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. So it's a 3-carbon compound. Fructose 1,6-bisphosphate gets converted into glyceraldehyde 3-phosphate and dihydroxyacetone and acetone phosphate. Okay, this glyceraldehyde 3-phosphate is a 3-carbon compound, and also dihydroxyacetone is also a 3-carbon compound. So the fructose 1,6-bisphosphate is cleaved into two 3-carbon compound. Okay, for every 1 molecule of glucose you will produce 2 molecules of glyceraldehyde 3-phosphate. Okay, right. So now this is generally catalyzed by enzyme called triose phosphate isomerase. I told you these are isomerase. They are isomers, right? Depending upon the availability and necessity, this DHAP would keep converted into glyceraldehyde 3-phosphate, and this glyceraldehyde 3-phosphate would further go for the stage 3 process for the conversion into the pyruvate. Okay, and I want to tell you one thing. Glycolysis is an aerobic process. 95% glycolysis in the cells is aerobic. Generally it takes oxygen and usage, right? So if the glucose molecule is broken down and converted into energy, only net gain of 2 ATP is produced from glycolysis. But the complete generation of energy from 1 molecule of glucose, it has to go for the Krebs cycle that happens in the mitochondria. Okay, so what happens is, in mitochondria there will be production of ATP, NADH and FADH. So where NADH and FADH would go for the electron transport chain that happens in the mitochondria for the ATP generation. So without any confusion, I want to tell you one thing. Glycolysis is an aerobic process which happens in all the cells. Okay, and this glycolysis in the presence of oxygen would get converted into pyruvate. Okay, the fate of pyruvate would be complete oxidation into carbon dioxide, okay, in the presence of oxygen, or it may either go for anaerobic process called as fermentation. It may either produce ethanol or lactate under anaerobic conditions. Okay, so this I want to talk to you in the early class itself, but now I thought it is must for you to understand, right? Okay, now let's go for the glycolysis of stage 3, where the substrate level phosphorylation would happen. Okay, so substrate level phosphorylation is an important step, being the first ATP is produced in the stage 3. Okay, so till now your glyceraldehyde 3-phosphate is ready, and dihydroxyacetone phosphate would be interconvertible. Okay, now glyceraldehyde 3-phosphate would enter the glycolysis cycle where the substrate level phosphorylation would happen. Okay, so this stage 3 is considered to be the energy producing stage. Okay, till in the first 2 stages there is no energy production. Energy production would happen only in the stage 3, where the glyceraldehyde converts itself into 1,3-biphosphoglycerate. Okay, and here the enzyme called as dehydrogenase is involved. So what is dehydrogenase? This dehydrogenase is important for the oxidation and reduction reactions. It will help for the oxidation reduction reactions. What do you mean by oxidation? Removal of hydrogen atoms is what you call oxidation. At the same time, when hydrogen atom is removed and when it is attached to the other molecule, that particular molecule is called reduced molecule. Reduction happens. Okay, and here what happens is the glyceraldehyde 3-phosphate would get converted into 1,3-biphosphoglycerate, where the conversion happens, and here the hydrogen atom from NAD gets reduced to NADH. So the enzyme called dehydrogenase is involved, and this NAD is present in the cells, and it is generally getting from, you will get NAD generally from the nutrition, okay, from the B complex like niacin and all, okay. And now this 1,3 biphosphoglycerate is converted into 3 phosphoglycerate, and here this is the important step in your glycolysis, because here this is the step where you produce the ATP molecule. And again I told you kinase enzymes are involved only for the conversion of your phosphate group from one molecule to other molecule. And here what they have done is, already you can see there is a 1,3 biphospho, here already you can see 1,3 bisphosphoglycerate, means you have got a phosphate group in first position and in the third position, right? And now this phosphoglycerate kinase would convert that phosphate group present on the first position to adenosine diphosphate, means it is transferring the phosphate group from the first position to the ADP. Thus ADP is getting converted into ATP, addition of phosphate, and this 1,3 biphosphate would lose this phosphate group in the first position, leading to the formation of 3 phosphoglycerate, okay. And here ATP is produced. So remember, this whole process occurs twice, because I told you glyceraldehyde 3-phosphate is a 3 carbon compound, glucose is a 6 carbon compound. So for every 1 molecule of glucose, you will be getting 2 molecules of glyceraldehyde 3-phosphate, and thus the whole process occurs twice, okay. So remember, here already we got a 1 ATP molecule generated, and here 3 phosphoglycerate is again converted into 2 phosphoglycerate, which is an reversible reaction, and it is again catalyzed by enzyme called phosphoglycerate mutase, okay. And now this 2 phosphoglycerate gets converted into phosphoenol pyruvate with the release of a water molecule, and this is catalyzed by enzyme called enolase, okay. And now here again the phosphoenol pyruvate is now completely reduced to pyruvate again, and here the kinase enzyme called pyruvate kinase would help for the conversion of phosphoenol pyruvate to pyruvate by transferring the phosphate group from the phosphoenol pyruvate to ADP, thus making ADP to ATP, okay. And thus here energy is again produced in this step. So at stage 3 process you have two types of completely 4 ATPs are being produced in the stage 3. As I told you, it's a 2 times process. It's in the stage 3 you have 4 ATP produced, and in the stage 1 you have already utilized 2 molecules of ATP in the first step itself, right? So in a stage 1 where it's an energy investment process, you have already consumed 2 ATP, and here you have produced 4 ATP. So when you totally balance it, the net gain for the glycolysis is 2 ATP, okay. For every 1 molecule of glucose, you will be getting 2 ATP, okay. So this is how the glycolysis happens, and this pyruvate later gets reduced to acetyl coenzyme A, and finally it will go for the Krebs cycle, where the complete oxidation would happen, and where CO2 is produced, okay. So this is the important glycolysis step that I want to discuss in this step. I will give you the detailed links with diagrams in the description box. Please check it out, okay. The enzymes related to it, the description about each and every enzyme, I will link it into the description box, okay. So this is the glycolysis cycle, and I want to tell you one more important thing. So what happens when the muscle is at rest, and what happens when the muscle is at exercise, okay. So this is the net reaction that I want to tell you. For the 1 molecule of glucose, you'll get 2 molecules of pyruvate, 2 molecules of ATP produced, 2 molecules of NADH and water. So this is the net reaction rate that you have to be aware of, okay. So what happens here, let's see. You all know that when you are in need of energy, like if you are doing some exercise and you are in need of energy, so glycolysis should happen, right? Then only the glucose in your body gets converted into ATP, and your energy is you will be regenerated with your energy, and you can perform the work, right? So what happens during exercise? So glycolysis is stimulated, where the glucose and the glucose 6 phosphate conversion would happen, and these are the things that happen when you are under exercise. This particular ATP generated would help for the muscle contraction, okay, during your exercise. So that is how the glycogen would also help for the conversion into glucose 6 phosphate, where the glycolysis is performed well during exercise. So at rest what happens? So at rest what happens? You all know that more and more amount of glucose in the body is stored in the form of glycogen, as I already told you. Okay, now you don't need the glycolysis to happen because it is at rest stage, right? At rest stage the glycolysis has to be stopped because you don't need energy during rest. You are just taking rest and all the glucose is converted into glycogen. Okay, now at rest what happens? The glucose has to never go for the glycolysis. So for that to happen, the glucose 6 phosphate, whatever it is converted, has to get converted into glycogen. See, here it's a negative thing that is happening. Instead of going for this cycle, the glucose 6 phosphate would immediately get converted into glycogen and stored in the body, so that glycolysis cycle is completely inhibited. And that is why even I told you that I want to tell you in the next class about the importance of hexokinase, phosphofructokinase and pyruvate kinases. These three important, they play a very important role in controlling the reaction of glycolysis. Okay, the rate of glycolysis, in the net rate of glycolysis, is mainly dependent on these three enzymes. Okay, and these three enzymes under rest conditions would help for the muscle to relax. Okay, so that the glycolysis would never happen in the muscle fiber. So these are the two important things that I want to discuss. Okay, so hope you are clear with glycolysis. Hope I have done a simple brief about glycolysis. In the next class I will talk about the further important metabolic reactions that happen in the mitochondria. Okay, so thank you and have a nice day.